Auzubillahiminash-shaytanir-rajim. Bismillahir-Rahmanir-Rahim. Today my lecture is about another kind of population in the soil, the protozoans. Protozoans are present in soil. They are the single-celled organisms. They are only visible through microscope. They are basically eukaryotic organisms. Eukaryotic means they have well-defined membrane-bounded structures. They have well-developed nuclei. These organisms are placed under the definition of soil fauna. Means when we talk about the soil fauna, then two major organisms are present in two major organisms come under this definition. One are the soil protozoa, and the other group is soil nematodes. So our next lecture will be about soil nematodes. So this group may be divided into 4 types of organisms based on the way they locomote. The first one are the flagellates. Flagellates are the protozoans that have flagella. For example, you see in this figure, this organism has 2 flagella. Similarly, this organism also has 2 flagella. Here you see 4, and here you see many flagella. So the flagella are present, and such organisms are known as flagellates. The other group are known as ciliates. As you can see here, 3 different types of protozoans. They have a number of cilia around their body. Cilia are small hair-like projections. They show a symmetric movement whenever the organism wants to move in a particular direction. Then there are naked amoeba. You see here the amoeba kind of organism. The naked amoeba don't have any shell around the body, and the protoplast is visible through naked eye. And similarly, there are shelled amoeba which have shells around their body. Now, the protozoa require water film for locomotion and feeding. So availability of water is a very serious requirement for protozoa. That's why protozoa can only show activity in the water-filled pore spaces present in the soil. So under the adverse condition in soil, when water is not available in the drought conditions, the protozoa make cysts. The cysts are highly resistant structures, and they are the survival structures under adverse conditions. Another character is reproduction of protozoa. Protozoa reproduce both by asexual reproduction and by sexual reproduction. The asexual reproduction is by binary fission. Binary fission means the cell divides into 2 by simple cell division, and the So in the sexual reproduction, the two compatible cells fuse together and make a zygote. Now we talk about the ecological importance of protozoa in soil. They play important role in the soil ecosystem. They are a part of the total soil respiration and soil biomass. They are present in all kinds of soils where water and organic matter is available. So whenever we take the total biomass of the soil, protozoa are part of it. Then they act as predators, and as a predator, they have a role in controlling microbial communities. They feed on bacteria and fungi and the minor organisms. The fungal feeders are also known as macrophages. So in this way, they act as a predator of smaller organisms, including bacteria and fungi. Then these organisms play important role in the soil ecosystem by providing nutrients through excretions. Their excretions act as a source of mineral nutrients such as carbon and nitrogen, and many microorganisms grow on there. The numbers and biomass of protozoa are highly dependent on the food supply. If the food is sufficient, the organic matter is available. If the quantity and quality of resources are good for the protozoa to flourish, then we will see higher number and biomass of protozoa. For instance, when the quantity of litter increase, it has been observed that the number and biomass of protozoa also increase. Another important factor that control the number and biomass of protozoa in soil is the moisture. As we have discussed that the moisture has a key role for activity of protozoa. Major activities of protozoa are in the pore spaces where moisture contents are available. Even they can move in moisture. So if moisture is not available, if there is drought, they may shift to their other phase of their life cycle, that is the making survival structures, resistant structures such as cysts. Then the aeration and pore spaces in soil also play a role. Land management practices can also affect the protozoal communities, may not be directly but indirectly, because land management practices have an impact on organic matter and moisture contents. That is why the land management practices may alter the population, because they alter moisture condition, because they alter organic matter in the soil. And organic matter and moisture are the key components that define the number and biomass of protozoa. And another thing that has been observed and that has been reported by many scientists who work on soil protozoa, they have reported that as the depth of soil increase, the number of protozoa also decrease. What is the reason behind? It has been hypothesized that probably it is the organic matter. It is the organic matter which decrease with increase in depth, and with the decrease in organic matter, the food supply is lesser to protozoa and number of protozoa also decrease. Although we can't say that protozoa can't survive in deeper soil, they have been found even at a depth of 7.4 meters below the surface of soil. So we cannot say that the protozoa are not available or cannot survive in deeper soil. They are present, but in lower numbers. Another thing that we can compare here is the use of land. I mean, what is the vegetation on land? The natural land cover contain more protozoa as compared to the ones that are manipulated or cultivated. Means the lands where there are grasslands, there are forests, there's natural vegetation cover. It has been observed that such lands have more protozoal population. While in cultivated lands, where different crops are grown, where different manipulations are done, plowing is done, nutrition is applied, different for example, different crops are grown and then they are harvested and then other crops are grown. So such areas are known as cultivated areas. So such area have lower number of protozoa. What's the reason behind? So we can see, because the lands are disturbed, that may be the reason why protozoa are lesser. Secondly, because of reduced energy inputs and soil exhaustion, means we take away a lot of carbon in the cultivated areas. We take away a lot of carbon. We harvest crops. That may be one of the reason. Another thing that is application of organic matter in the soil. It has been observed that when we apply organic matter to soil, the number of protozoa increase, its biomass contribution also increases. What's the reason behind this increase? Why the application of organic matter may increase the protozoal number and biomass? It's very simple, because the numbers and biomass of protozoa depend on the availability of food and nutrition, and they use organic matter for that. Then there are then the number and biomass also depend on the plant residues. There are two things that I have to mention here about plant residues, how plant residues can affect the number and biomass of protozoa. If the residues are placed on the surface, they probably have no significant effect on the number of protozoa, or the number may be reduced. The reason may be that on the surface there is more aeration. The residues are more exposed to the fungal attack, And however, when these residues are buried in this, below the soil surface, it has been observed that the protozoal population increased significantly. Then another thing that I would mention here is the application of inorganic fertilizer. It has been experimented, and it has been observed that whenever we apply inorganic fertilizer to soil, the protozoal population increase. What's the reason behind? Probably when we apply fertilizer, it increases the plant vegetation, it increases the organic matter, and that may be the reason of increase in protozoal population upon application of inorganic fertilizer. Now, how the protozoal population is distributed in the soil, which are the areas where we find more protozoal population. One thing is very clear. One thing on which everybody is agreed is that the distribution of protozoa in soil is highly variable. It is not uniform. Even it varies, and it depends on the areas where plants are growing. For example, the protozoal population and density is much higher inside the plant rhizosphere, where there is more organic matter. Similarly, the irrigated areas where water is provided for plant growth and development, such areas have more protozoal population. What's the reason? The reason is again the same. Because we increase water, the moisture available is higher, and secondly, the plant cover is also increased. So in these two things, moisture and organic matter, they favor the increased population of protozoa. Then grasslands and forests have more population because such areas are undisturbed and they have more organic matter. Then deserts and extreme environments have small population because of lower availability of moisture and organic matter. The protozoa react very quickly to the environmental disturbance. That's why protozoa are used as an indicator in laboratories to environmental disturbance or toxicity indicators. So protozoa can be used as an indicator of environmental degradation. So how we study protozoa? Now here we discuss the different methods that are used to study the protozoa. One is the direct observation of protozoa in soil extracts. Using microscopes, we can see protozoa using microscopes. We extract the soil, and then we see it under the microscope. However, the drawback is that in this extract we also have some soil particles that may hinder the accurate estimation of protozoa. So to avoid this hindrance, we can perform gradient centrifugation, in which the protozoa are separated from the soil particles, Then we can also culture the protozoa. In this case, we make a number of cultures, and then we use most probable number technique. Then another way to study the protozoa are use of molecular methods. For example, using DNA markers. In this case, we extract DNA and then use specific primers to amplify the specific region. Specific means that they only amplify the particular region of the protozoa, not of other organisms. So then these amplicons can be amplified. In some other signature molecules are also used for quantification of protozoa, for example, quantification and characterization of protozoa. For example, lipid and protein markers are there, so we can use them. Thank you very much for your attention. If you have any question, you're welcome. You can ask questions through KCMS. Thank you very much.